This method extends DNA nanotechnology from the source materials of linear oligonucleotide DNAs into small circular DNAs. The main advantages of this technique are that it is simple, robust, and affordable for most labs.
For circular DNA purification, add 20 microliters of formamide to a solution of freshly prepared intact circular DNA to a final volume of 140 microliters with mixing. Load 16 to 20 microliters of circular DNA solution to each of 7 to 9 denaturing page gel wells. Mix 2 microliters of loading dye with 8 microliters of the corresponding precursor linear DNA and inject the mixture into a separate reference well. Then run the gel for about 2 hours at 10 volts per centimeter, stopping the run when the xylene cyanol FF can be observed about 2/3 down the gel. Wearing rubber gloves and goggles, transfer the gel onto a fluorescent thin-layer chromatography plate and use a razor blade to cut off the target gel bands as shadowed by the UV irradiation. Place the gel bands into a 2 ml microcentrifuge tube for drying. Then mash the dried gels into a paste and add deionized water at twice the volume of gel for overnight shaking at room temperature. The next morning, filter the mixture into a 2 ml microcentrifuge tube, rinsing the sides of the first tube with a small volume of deionized water.